Awesome, yeah, so I've been giving this presentation for a couple years now at the Matsu Salmon Science Symposium virtually, and I guess I'm just curious with like a show of hands, how many of you guys have seen iterations of this work? Cool, a couple folks, awesome. Yeah, so my name's Taylor, and I'll be giving sort of the last installment of my master's research while being a student at UAF. And I've been working on this with my advisor, Dr. Jeff Falke, as well as co-authors Kevin Kappenman, Chrissy Dunker, and Matthew Blank on this Chapter 2 component. And this project was funded by the U.S. Fish and Wildlife Service. I'd also like to acknowledge that this research has been conducted on the ceded and unceded lands of the Lower Tanana, Dene, Dena'ina, and Notna people of Alaska, as well as the Blackfeet and Crow people of Montana. And I'm grateful for their past, present, and future stewardship of these habitats and aquatic resources that I will be talking about with you today. So to begin, this research is spurred by the issue of biological invasions, which many of us are familiar with, their negative impacts on our native ecosystems. But we're also realizing that invasions can act as really useful learning opportunities to study invasion biology and evolution, and how invaders often change really rapidly, and they're able to change in ways that often benefit their dispersal because it benefits them to move and expand into new habitats, such as longer-legged toads in Australia. And so this just highlights the importance of studying invasions while also trying to manage them to better predict and prevent future ones. A prime example of a successful invasive species that a lot of us are familiar with are Northern Pike. For example, in the lower 48, they've spread really rapidly outside of their native range. In the orange here into all of these maroon regions where they negatively affect native fish communities through predation. They're also native to a lot of the state of Alaska, but were introduced into South Central where they're not native. A lot of us are familiar with the spread and impacts of invasive pike in South Central, where they predate upon our native fishes, especially salmonids. There's been quite a bit of research, especially at UAF, to try to understand what might be driving the invasion success of pike in South Central by comparing invasive and native populations in Alaska because they're so close. For example, in Chapter 1 of my master's research, I did some physiological comparisons, some diet comparisons, and found that invasive pike had much better body condition and faster growth rates than their native counterparts, likely because of this abundant food resource. We're also learning that pike might be able to utilize Cook Inlet as this migration corridor, and so it just highlights the importance of understanding, how quickly they're dispersing, and they're in such good condition. So trying to figure out ways to stop them from spreading further. And one way to do this is this idea of selective fragmentation, where you design barriers or filters that still allow for strong habitat connectivity for your native species while reducing the spread of your invader. And so there are a lot of different ways that you can think about this. And whenever we think about our native salmonids and comparing them to those of pike, salmon are very metabolically active and will readily ascend waterfall barriers to access spawning grounds, whereas pike and other associates like this muskie just aren't really known for their leaping ability. So management agencies are considering the installation of short waterfall barriers that might be able to fit in this sweet spot between pike and salmon leaping abilities. But before we begin kind of moving forward with this, we first have to figure out the maximum leaping ability of pike because there just hasn't been a systematic assessment. So to do that, you first have to consider what affects fish leaping ability in general to figure out their maximum. And so there's quite a few factors that influence fish leaping ability. Some physical factors include the height of the waterfall structure, the plunge pool depth that they're jumping from, water temperature, and the flow rate coming up and over the barrier. Some biological factors specific to the fish that influence their leaping ability include their size, their body condition, how healthy they are, potentially their individual metabolic rates and growth rates. So to quantify the maximum leaping ability of pike, I directly manipulated these three factors in a large-scale experiment to see how they affected pike leaping ability while also quantifying these individual biological characteristics to capture that individual variation. And it's really important because when we think about our invasive pike in Alaska, they're in a lot better shape than a lot of populations because of that abundant food resource. And so ultimately, I built predictive models with this data to test and potentially design and test barriers in South Central as well as elsewhere pike are invasive. So this work was conducted at the Bozeman Fish Technology Center in Montana. We had this great partnership with the U.S. Fish and Wildlife Service. We were able to utilize this brand new flume structure that they have developed at the facility for the leaping experiments. The pike were also collected from Montana and Fort Peck Reservoir and trucked down to the technology center where they were held in a large flow-through tank and survived pretty well on a diet of live rainbow trout. So back to that flume. This is kind of a schematic diagram of what it looked like. We've got this large sump down here, and then water gets pumped up into this large PVC pipe into the headwater pool over on the right there, and then the water flows down the flume into the tailwater pool and then the sump again. So it creates this really nice enclosed system that we're able to control flow rates and water temperature in really well. And so I also divided the flume into 3 major sections so that I could run 3 leaping trials simultaneously. Kind of zooming in on what one of those sections looks like. I've got a lower pool down there where I place the pike. I use tongue and groove boards for the barriers. And then the upper pool there has a pit antenna in it, and each of the pike were tagged with pit tags so that I could detect when and which individuals jumped up and over the barrier during the trials. In terms of the experimental design, I first ran an experiment looking at how height and depth affected pike leaping ability, testing 16 different height and depth combinations, pretty factorial design here with heights ranging from 10 to 65 centimeters and pool depths ranging from 20 to 80 centimeters. I repeated each treatment 3 times each at a moderate flow rate of 27 liters per second, and I used 2 pike per trial for a total of 6 pike exposed to each treatment to just increase that range of biological variables. And in terms of my variables, I was quantifying passage success as a function of height and depth, with water temperature held constant at 11 degrees Celsius for all the trials and that flow rate constant at 27 liters per second. And down there you can see those pike covariates that I quantified as well. I was also able to run a second experiment looking at how height, depth, and flow affect pike leaping ability. Here I took a truncated set of the initial 16 combinations that had a wide range of passage success rates and repeated them at a low flow rate of 11.5 liters per second and then a high flow rate of 43 liters per second so that I could then compare the passage success rates at the same height and depth combinations to that moderate flow rate of 27 to kind of see how pike also respond to flow rate. In terms of the experimental procedure, I would place pike in the flume and allow them to acclimate for a few hours, and then they'd volitionally ascend the flumes overnight if they could. And then I'd scoop the pike up and return them back to their holding tank in the morning. And once all the trials were completed, I euthanized all the pikes so that I could collect those biological metrics, their age, their sex, their growth rates, body condition, and metabolism. And ultimately, I used logistic regression modeling to include these biological and physical predictors to see which predictors best estimated pike leaping ability. So now we can jump into some of the results from the pike that I used in the study. There were 55 total, 44 were males and 11 were females with 98% maturity. In terms of those biological metrics, we didn't have a huge range in pike fork lengths. They were all, adults. We didn't have a huge range in metabolic capacity or body condition measured as dry lipid content, but we did have quite a range in pike ages and growth rates, which will be important later on. Now we can get into the results from that height and depth experiment, so those 16 combinations. The combination with the highest passage success was a 10 centimeter drop with the 30 centimeter pool depth with 84% passage success. And then the combination with the highest, or the tallest drop with 1 successful leap was the 40 centimeter combination with a 65 centimeter pool depth. However, when that pool depth was reduced to 40 centimeters, we saw 0% passage success. And just to confirm as well, that 65 centimeter drop had no passage success as well. Then looking at results from the height, depth, and flow experiment, that same 10 centimeter drop with a 30 centimeter pool depth had the highest passage success rate at the moderate flow rate. And then the 40 by 40 combination had 0% passage success across all tested flows. Now we can take a look at the model selection results. So this was, including all those predictors together, the final model ended up being passage success as a function of barrier height, pool depth squared, an interaction term between fork length and pool depth, and standardized growth rate. However, individual pike body condition, metabolism, age, and flow rate coming up and over the barrier were not significant predictors based on our data. So now I'd just like to walk you through what some of those, what passage success looks like as a function of some of those predictors, their effects. So we can look at barrier height here. We got passage success on the y-axis and barrier height on the x, and what we saw is barrier height increased, we saw passage success decline at a cutoff of around 40 centimeters. Next, looking at pool depth. So here we saw as passage success, or as pool depth increased, we saw passage success also increase, but sort of to this optimum, where once we surpassed this optimum pool depth, we saw passage success decline. Next, we can look at that interaction between pool depth and fork length. So we've got this graph here, again showing pool depth on the x-axis, that moderate pool depth having the highest passage success in the middle there. Those different colors just represent different barrier heights. So 10 centimeters has a really high passage success and it declines as you get down to 40. But the next layer here, these three panels represent different pike size classes. So we've got small, medium, and large pike. And what we saw here, this interaction, was that as pike got larger, they needed deeper and deeper optimum pool depths to ascend similar barrier heights as small pike. And this makes sense because, larger pike need more space in that pool to orient vertically and get a good, well-timed leap up and over the barrier. So that last significant predictor was growth rate, standardized growth rate. So it wasn't a function of, we took out the effect of age, and this was still an important effect here. And it's likely that we're able to capture this because we did have such a wide range in pike growth rates, lots of individual variability. And it's interesting to think about because it's likely not that individuals that grew faster somehow were able to get up and over the barriers better. It's likely that there was some sort of accumulation of individual benefits that facilitated growth as well as leaping ability. And so when we think back to, the grand scheme of things, we're thinking about pike in Alaska, not really pike in Montana, but I compared the growth rates between the two populations of pike in Alaska and those that I used in the leaping experiments, and I found that Alaskan pike had 2 times faster growth rates than the ones used in leaping studies, but this resulted only in about a 4% increase in predicted leaping ability, so it's interesting to think about the biological relevance there. So to conclude, we captured a pretty wide range in pike leaping ability here. We were able to see that they could ascend 40 centimeter barriers, but this is a lot less than what adult salmonids are capable of, 70 centimeters to a meter. But that 40 by 40 combination was impassable across a wide range of flow rates. And it ended up being that those physical barrier factors of height and depth had a stronger effect than biological traits. But we weren't able to test for effects of water temperature or higher flows or seasonal motivation in individuals. Ultimately, we found a good starting point for barrier designs, but some in situ testing will be really important in these ecosystems to see if they, before they should be widely implemented. And so I'm out of time. Thanks everybody for listening, and yeah, happy to take any questions and look at all those fantastic people that helped me along the way.
Taylor, any questions? Got time for one or two.
Hey Taylor, I was just wondering if in your discussions about this work with managers, there was consideration for juvenile salmon being able to migrate, obviously to overwintering habitat or something, as opposed to just adult salmon leaping abilities.
Yeah, that's a really fantastic point, and there's been a little bit of research out there looking at what juvenile salmon can ascend, and some coho and steelhead studies of juveniles that are a little bitty, they can actually ascend 40 centimeter barriers with some success. So we're kind of right on that cusp of maybe facilitating some juvenile movement. We definitely don't want to prevent that as well because that's really important. So, and there are also, barrier designs that could incorporate, juvenile movement, like beaver dam analogs that have, really small pockets that they could move in, but at the same time, you want to make sure that there aren't juvenile pike in that system too. So, something to consider for sure.
Okay, one more from the back, and then we'll go to the next talk.
Hey there, quick question about pool depth. It was surprising that increases in pool depth past the asymptote seemed to decrease success. That was surprising to me. Any ideas about why that might be?
Yeah, so it's interesting, and especially whenever you think about that interaction with fork length, we saw really, really strong declines in when that pool depth was really deep, especially for small pike. And it might be sort of a behavioral thing where, small pike, they're in this really deep pool. There's not a ton, it's a lot less turbulent than a really shallow pool, so they don't have as much motivation to leap up and over the barrier. But we still saw like quite a bit of, once the pool depth was deep for those large pike, they still wanted to get out, likely because they're more sensitive to all of that turbulence still occurring in the pool. Because they're larger, they aren't able to find those little pockets of non-turbulence to hide in. That was something really cool to see.